Good afternoon, everybody. Thank you for having me here in the meeting today, and also thank you for coming to the meeting on Saturday. When you're supposed to rest, right? So thank you for joining the meeting. And my topic today is the assay technology and performance of our SARS-CoV-2 IgG assay. In May, early May, we actually launched our SARS-CoV-2 IgG assay in response of the needs for the testing for this COVID-19. And our assay is a IgG assay just to testing the IgG antibody, and this is based on the chemiluminescent microparticle immunoassay. Right, and this assay is a qualitative assay, and it's supposed to be used over analyzer like Architect and Alinity. The assay is the two-step assay. I won't go into the details about the reaction, the mechanism of the assay due to the interest of time. But the total processing time for this two-step assay is 29 minutes, and it can be run on the automatic analyzer. So I'm sure you are quite familiar with I'm sure you are quite familiar with this structure of the SARS-CoV-2 virus. There are four structured proteins of the SARS-CoV-2 virus. This spike protein on the surface looks like a spike, and it gives the appearance of the virus as a crown. And then there is an envelope protein, membrane glycoprotein, and also nucleic capsid protein. So this spike protein and the nucleic capsid proteins are the two immunodominant proteins on the virus. And our assay actually used the nucleic capsid protein in our assay design to capture the virus antibody. So a little bit more about the proteins, right? The S protein and the N protein. The spike protein or S protein is responsible for the viral attachment or the binding of the viral with the host cells. And also it will help the virus to fusion and entering into the host cells. And hence, it helps the transmission. But for the nucleic capsid protein or N protein, it's responsible for subgenomic viral RNA transcription and the viral replication and the assembly. Actually, there are a lot of studies ongoing on the S protein and the N protein. And N protein is found to be the most abundantly expressed immunodominant protein. And in regards of the kinetics of the antibody response to the virus infection, there are still studies ongoing. But at this moment, right, so far, actually, the reports said that the kinetics of anti-N response was reported to be similar to the anti-spike. Although actually some of the reports said the N response might appear earlier than the anti-NS, right? And in the context of the SARS-CoV-2, actually, when we're looking at the antibody, the neutralizing antibody becomes very important because neutralizing antibodies are the type of the antibodies that can neutralize the virus and then can stop the virus from entering into the host cells and infecting the host cells. That's why people always want to find out when this patient produced the antibodies, whether there are neutralizing antibodies. So the neutralizing antibodies at this moment, right, is believed to be predominantly from the anti-S antibodies. However, a lot of evidence, I'm going to show it later, actually shows that the N protein designed assays also showed equal and sometimes better correlation to the presence of the neutralizing antibodies. This is a couple of reasons why our assay actually use the N protein in our assay. So this is actually the study that demonstrate the good correlation of our assay, our architect assay, against the activity of neutralizing antibodies. So in this study from the Helsinki, Finland, right? The authors actually evaluated 6 SARS-CoV-2 immunoassays, including 5 IgG assay, of course, including our assay, architect assay, right? And then the authors actually compare the results from these 6 assays against the neutralization test result. So what they found is they actually included our architect IgG assay, disoring assay, Euroimmune IgG assay, and a few other rapid test assays. So what they found is in terms of the positive results that are produced by the neutralization test, right? Our IgG assay actually has agreement of 80.5%. And for the negative samples, that with the negative results on the neutralization test, our agreement is 95.1%. And this set of the results is the best among all the IgG assay. And this is quite interesting because our assay actually used the nuclear capsid protein or N protein. But disoring assay, they use the S1, S2 is the spike protein. Actually, the S1 unit and S2 unit of the spike protein. But yet, in relation to the correlation between the assay result and the neutralization test result, our assay, although it's an N assay, has a much better correlation against the spike protein designed assay. So this is regarding the performance of our assay in relation to the assay sensitivity and the specificity, or the positive agreement or negative agreement, right? So in terms of sensitivity, our assay has 100% sensitivity for the specimens above 14 days post-symptom. And, of course, at the very beginning of the infection, we can't detect IgG antibody. And then with the infection actually advances, more antibodies will be produced. So for the samples above 14 days, we can detect 100% of the IgG antibodies. And in relation to the specificity, we have tested our assay on over 1,000 samples, including other respiratory illness samples, right? And our specificity is 99.63%. On top of the specificity, we also have done a very thorough cross-reactivity check on other related specimens, including the specimens like influenza A, influenza B, or influenza, the patients with the influenza vaccine, because a lot of people actually has the vaccination from influenza, right? And also, the samples are from like the RSV, because all these patients will present a very similar clinical symptom like the SARS-CoV-2. So very importantly, we want to make sure there is no cross-reactivity to those patients. And of course, other virus samples. And all together, we actually have validated against 36 categories of related specimens, and the results are quite good. And this is the most robust evaluation out of all the antibody assays on the market. So I just want to, emphasize a little bit more on the assay performance because, although we have developed this assay in a short period, actually shorter than what we ever did, what we ever done for other assays. But yet, we still strictly follow the CLSI guidelines in our assay development. And especially in the assay validation, we have validated over 1,000 samples on the specificity and also other respiratory illness. Because the number of samples in the assay validation is really important, because if you validate more samples, it will give you a higher confidence to make sure the results are responsible. So that's why when you see the 95% confidence interval here, actually you can see the 95% interval here is really very narrow. That's because of the large amount of the samples we have validated. So that's why, when you look at the assay performance sheet, you really need to be careful, because what it tells you probably is not what it reflects in your hand. I just take this performance sheet from a rapid test as an example. So at one glance, when you see this performance sheet, actually this assay shows a very, very good specificity, 100% specificity when they test the negative samples, right? And also the assay has a good sensitivity, 97.1% when they test the confirmed samples. However, if you do a deep dive into the performance sheet, you will see that they only use 50 samples in their specificity validation. And with these 50 samples, they have 100% specificity. But the question is whether, such a small amount of samples will give you an assurance or confidence that the assay will reproduce the same results in the user's hands, right? And also, this assay actually didn't give you the 95% confidence interval. So you don't know, whether it is, you can always repeat the results as 100%. And also, if you look at the performance sheet, they haven't validated the cross-reactivity against other related samples. So, if the assay is used for example, for influenza or for other coronavirus like the common cold or other virus, whether there is any cross-reactivity, you will not know, right? So that's why assay validation is really important to make sure that the assay can perform as we claimed or as it is intended. And again, this is actually about the quality and performance of the assay and why it is relevant to your day-to-day work or to your day-to-day clinical utility. So I'm sure you all know these two parameters called positive predictive value and the negative predictive value. So this positive predictive value, or the PPV, is actually the percentage of positive tests that are true positive in a given population, while the NPV is the percentage of negative tests in a population that are true negative. So that means if you have 100 positive samples, if you have 100 positive results, and if only 80 out of these 100 are true positive, then the PPV is 80%. And also for the negative, it's the same, right? So this NPV and PPV, they can be calculated from the assay sensitivity, specificity, and the prevalence of disease in the population. And this actually gives you two examples for a population with the COVID-19 prevalence as 4, which is maybe a typical prevalence, 4% prevalence. And if you use test A with a sensitivity of 95% and the specificity of 95%, which actually looks quite good because both are 95%, right? But yet, when you calculate the PPV and the MPV with the prevalence of 4%, then you will only have a PPV of 44% and MPV as at 99.8%. So what does that mean? It means if you have 100 positive results, you will only have 44 true positive results, while 56 out of 100 are false positive results. So it means, when you tell your patients or even tell your colleagues, your healthcare worker colleagues, right, you have a positive antibodies, and then they thought they have a positive antibodies, but yet these positive results may be false positive. So, they will have a greater risk when they're exposed to the infection or the virus. But, if you use a high performance assay like our assay with the sensitivity of 100% and specificity of 99.63%, the PPV would be 92% and MPV would be 100%. That means there will be only 8 out of 100 positive results are false positive, and none of them will be false negative. So, in terms of the assay sensitivity and specificity, it's not only about the numbers. It's real, impact to your day-to-day work. And this is actually the real-world experience from our users. So this is a publication published by the University of Washington. The doctors over there actually evaluated our assay in relation to the sensitivity and the specificity. So they evaluated more than 1,000 pre-COVID samples, and they found the specificity of our assay is 99.9%, which is even higher than what we claim. And also, they found from these almost 700 serum samples, which are confirmed by the PCR, right? And they found the sensitivity is 100% for the samples above 17 days post-symptom. So this is actually the real-world data from our users. And a little bit more about the real-world data. So the University of Washington data I just shared in my last slide, and also the UK Public Health. So this is actually the government data, right? The UK Public Health also evaluated our assay as well as Roche, Diagnostic, Siemens, almost all of the serology assays on the market. And again, they found our specificity is very high, 100% over almost 400 samples. And the sensitivity is a little bit low here, but actually all these reports, the UK public health reports are available on their website. So if you look at all their reports, actually our sensitivity, although it's not 100%, but this is the best among all the assays, other like Roche assay, Siemens assay, or Diagnostic assay, their sensitivity are all lower than our assay. And this is from the Washington University. And again, the specificity is quite high. And this is a more recent publication from University of California, San Francisco. And the specificity is also close to 100% over 1,000 samples. And the sensitivity is 99% for the samples above 22 days post-symptom. So, these are a few publications already available, to evaluate our assay performance. So this is something probably you can refer to. In relation, in addition to the assay performance, I'm sure because you come from the hospitals, the labs, so you are also very interested in the laboratory productivity or the operational efficiency, right? So if you use the automated serology assay, actually it helps a lot, in the testing volume, because our assay has a processing time of 29 minutes, and it can achieve the throughput of 200 tests per hour. So it will enable a large throughput to satisfy the high demand of the sample volumes. And of course, because the instrument is already there, so you can just plug in the assay and introduce the kits. So it's in relation to the friendliness of the use, it's also quite easy. And if the instrument is connected to the automation system, like the check system, it will even automate the pre-analytical part. So you don't have to do the pre-analytical if you have an automated machine system. And this will help you, to reduce the potential errors as well as the biohazard risks. Because recently, WHO and the IACC actually issue, the biosafety, recommendations on the sample treatment. And if you have an automation system and automated instrument, it will actually reduce the chance that your lab personnel are exposed to the virus. And of course, if you are using a more complex algorithm, and for example, you want to confirm the assay with a supplementary assay, like PCR assay or other antibody assay, then if you have this informatics solution automation, then it will be configured to do auto reflex. So that will save you a lot of sorting and the time from your lab personnel. Now it's about the utility of the assay, the potential of this antibody assay in the real world use. So the antibody assay, one possible role for the antibody assay use is to identify those infected people who may already produce the immune response and potentially the immunity. So this could include the people, the high-risk people like the healthcare workers like yourself, or the essential workers, all those vulnerable populations like those old folks or those immune compromised people. So when you do the antibody detection, this might help. And another possible role is the seroprevalence study. So with the seroprevalence study in a community or even in a larger population, you will be able to understand what is the prevalence of the population, and you can estimate the real fatality rate, and also you can understand what is the transmission pattern, and eventually, of course, we all wish we can achieve the herd immunity, right? And then the third possible role for the antibody is to help the convalescent plasma therapy. So with the antibodies, it might help you to screen the donors, to see whether they have enough antibodies to be used for the therapy. And it can also help monitor the patients who receive the convalescent plasma to see whether their antibody will increase and whether their condition will be improved. And also, one other role for the antibody in the future probably will be to help the companion diagnosis. For example, if we have new therapy or if we have vaccine developed, then we can use the antibody to see whether the therapy or the vaccine can work to produce the antibodies. That's all from my presentation. Thank you very much for your time.